So earlier this week, we had this lecture on bioinformatics, right? And we talked about how we could predict structures from sequences. And today we're going to do a little bit the inverse game. We're going to talk a lot about which structures allow to see which sequences. And this is linked to protein folds, which folds are allowed or not, related to stability. And then we'll go a little bit into how these folds actually form. So talk a little bit more about folding or unfolding. You have already covered quite a few of these elements in kind of distinct lectures, right? Early on, you talked about secondary structure, why these were favorable energetically and entropically, right? Hello. You can take a... Yeah. Right, so we already, in many lectures, you've covered elements of these things. And specifically the last part, it's going to be reminiscent of the lecture that Burke-Hess gave on kinetics of folding, but we'll talk here about thermodynamics of folding. So I'm just letting you know because when you start looking at all the lectures together when you study for the exam, you'll see that it's difficult for us to order things in an order that necessarily makes sense because everything is kind of intertwined. So it's good if you know what links to what and in which way. Does that make sense to everyone? You're following? And then next week, I think you have your last lecture. And there you'll talk about, is that right? There's another summary session with questions and things. I think that Eric will lead in the also. But the last lecture is next week. And then you'll talk about drug design, and this is really kind of the most common application to biophysics. And all these principles that we've been talking about that sometimes maybe are a little bit dry, and you'll see how they get applied to a real case. Okay, so today, like I said, we'll talk about fold and why they're stable. And we'll see how this is related to statistics. So why certain folds are common, why certain are uncommon, what governs this, and how this is related to their sequence, and so the properties of the amino acids that make up these sequences. As I said, then we'll talk about transitions and how, now if we have different protein domains, do they fold all at the same time? Does one influence the folding of the other? And we'll touch a little bit upon other things than folding, which is transitions also between functional states, so bound, unbound, or that sort of thing. Does that look good? You don't have a choice anyways, now you're here. I don't have a choice either. Okay, so this is the mystery that you need to think about for a minute. Do you guys remember how many genes we have that encode for proteins in humans? The rough number? Do you guys remember DNA was really a large number of base pairs, and then how many genes did we have, roughly? Remember? 20,000, yeah, exactly, 20 to 25,000. So this means we have 20 to 25 different thousand different proteins that perform some kind of function in our body. Actually, something that's kind of interesting is that you might think this is a lot, but there are organisms that seem not very interesting, like the Christmas tree. They have 10 times that. And it's kind of interesting that, biology just is a bit random, right? So why does the spruce tree have 200,000 genes? Why do we have 20,000? And why do bacteria have a very low number? So this we don't know. Anyways, what we do know is this kind of famous quote by Chothia, 1,000 folds for the molecular biologist. This quote is a little bit old, so now we know that it's more 1,500. But still, if you compare 20,000 to 1,000, you have a good order of magnitude difference between that. So this means that amongst the 20,000 proteins, there are many that have a similar fold, even though they perform something very different. Why is that? Does that seem logical? To me, initially, before reading the Finkelstein book, it doesn't seem necessarily logical. We could imagine having 20,000 different proteins, right? Or at least different. They are different, but different architectures and folds. So why do most sequences fit a few folds? That's what we're going to talk about quite a lot today. Even worse, actually, out of these 1,000 folds, only 20% actually are very populated. So many, many proteins, 80% of proteins take up 20% of the folds only. So this means 200 to 300 folds to kind of fold 20,000 sequences, right? So now we're really kind of 2 orders of magnitudes lower. We can compare to the other bioproteins. RNA also has a large variety of folds, but DNA, you know, right, mostly has one fold. It's the double helix. In fact, there's 3 different types of helices, but mostly one. So it's kind of weird that DNA folds into one fold, but proteins fold into many folds. If you do think about function, it kind of makes sense, because DNA, what's its function? Exactly, it stores information only, so it doesn't need to be kind of finely structured into something. It just needs to be in a way that we can separate the 2 strands, and then we can translate into RNA and then get proteins. So maybe that's one of the reasons there's no pressure on DNA to fold into something else. But for RNA, which you haven't talked a lot about in this class, and I personally don't know much about it, but RNA actually was thought initially, transfer RNA, to only be a vehicle for transforming DNA into proteins, but it turns out there's actually many different types of RNA, and they fulfill different types of function. And you'd have to take a class on RNA, I guess, to find out more. But that's the story. So proteins, few folds, but much more than DNA, about 300 that fit 20,000 structures. Okay, so why are they similar? Can you guys think of a few reasons why we might have only a few folds? There's the answer behind me, but we can maybe talk about it for a minute. What is evolutionary divergence? This is very related to this week's lecture, the one on bioinformatics. You remember? No? Common ancestor? No? Okay. So the idea is that maybe we originated, all of us, or all living beings, originated from one organism that had proteins, and this is the reason why our proteins look the same as the one of the mouse and of the Christmas tree, is because we all came from the same organism. And so evolution has acted on a few folds, and this is why folds are a bit the same in all proteins. That's a plausible explanation, right? What is convergence of fold? This second one in your mind?
Could go membrane proteins in that category?
Mm-hmm.
For audiences, for example, doing that channel or something.
Right, exactly. So,
membrane proteins have the same structure.
right, ion channels, for example, have to conduct ions, and they have to be in the membrane. So you're saying, I guess, they need to have a hydrophobic outside and a hydrophilic inside so that they transport ions. So maybe there's not so many ways to organize protein chains into something that has the same function. Right. And it's the same, by the way, if you think about, binding a hydrophilic compound, then you need to have a soluble protein. So then you have to have a hydrophilic outside, a hydrophobic core, and a hydrophilic inside to bind the protein. So that's the idea behind functional convergence. And then the other thing, which is the reason that we're going to be the most interesting with today, is the physical restrictions that are put on folds. And this is called here a limited number of possible folds. So if we believe that physics govern the world, which I think we do, then maybe physics puts some restrictions onto what is possible to do with a protein chain. So which one is it out of these three? Yeah, a little bit of everything, and then, as always, it's going to depend on the specific protein. But a little bit of these three elements are going to be important to determine which folds we have to perform which function. So this we can only do because we have enough structures. So thanks a lot for structural biology. They have given us enough structures to try and start building a classification of what folds exist, right? We can only do this if we have enough folds. I'm sorry for the quality of this. It looks better on my screen. It's in the Finkelstein book. So since the end of the 70s, early 80s, we have enough structures to classify. And this would be a representation of the main classes of proteins. So what can you see on here? The alpha helices are represented in red, and the beta sheets are represented in green. I think on here you should see 3 families, so-called architectures, right? Alpha only, beta only, and mix of alpha and beta. So basically this means that there's, just to clarify, we're talking about globular protein here, not membrane proteins specifically in this lecture. We'll come to membrane proteins once in a while, but I'm mostly talking about globular proteins, so the ones that live in solution. So this is architecture, and then the arrangement of the architectures will build up the topology. And as science has it, once you start having enough representative of structures, then people become interested in classifying. And classifying is not straightforward, so this means that there's going to be different ways to do it. And these are 2 main databases that do that. So this is a little bit like we talked about databases as a part of bioinformatics, right? So this is a bit also where bioinformatics kind of meets physics. So this first database is called CATH, for Class Architecture Topology Homology. And this database is purely automatic. So there's an algorithm that takes up a structure from the protein data bank, and then it automatically classifies it into this kind of tree thing. So we said before, right? Class is whether it's alpha, alpha, beta, or beta. This is not so difficult to classify for an algorithm. And then you have the architecture. So if it's alpha and beta together, then there's different types of architectures. I don't know if you have gone through these folds. Like the names, they don't really matter, but they're like biologists will know these things. So if you tell someone that it has a sandwich fold, they'll be able to know that it's a beta sheet in the middle and then helices around them. You haven't talked about this before, no? So Tim barrel roll, these are different types of architectures that biologists will know the names of, and you don't have to remember them. And then we go into topology, and topology is then, so from the sandwich, which was a beta sheet with 2 helices, then you can have beta sheet that is made by how many beta strands and how many helices on either side, right? So this would distinguish, for example, this topology from that topology, right? And then homology, we try to put in also something from sequences. So sequences that are homologous, then we can further distinguish these topologies into different families of homologs. And then we have the completely opposite idea. SCOP is a highly curated manually database. So I don't know how cool you think this is. It's like people downloading the structures and annotating manually and classifying into different folds. So it doesn't obey in the same way kind of this tree-like structure, because it's more based on intuition and what people know of biology. And this actually is useful sometimes. Like, for some reason, maybe there's an alpha helix on some structure, and you know because you're a biologist and you've studied this protein a long time that this helix is not important for function. So you don't put it into your classification. That's the philosophy behind this database. It's much fewer structures just because it's manually curated, but it's good to do biology because you won't have anomalies that you would get from an automatic classifier, basically. Okay, so the way then Finkelstein introduces this is what's the most exciting question for a biologist is if you can understand evolution. And so does this classification then help understand evolution? If you think about globin fold, so if I go back just a couple slides, globin is one of the most found type of structures. It's purely alpha helical up there. And several globins will form, for example, hemoglobin, which is the protein that binds oxygen, right? You're aware of this, and it's very useful for life. And so we know that there's going to be pressure on this globin fold to perform a specific function. The example that's given in the book is why does the llama globin bind oxygen better than the horse globin? Where do llamas live? Where do llamas live?
So in the higher regions.
Okay.
Near air.
So less oxygen pressure, which means that you need a protein that binds oxygen tighter than the horse. Yeah? What about fetal hemoglobin? Fetal hemoglobin, so in the fetus, has a, say again? Why? Exactly. So this is a kind of sad story in a way, but the fetus has to steal the oxygen from the mother, and the mother has like the regular hemoglobin. So then the hemoglobin of the fetus must bind oxygen tighter. But you also, I guess, don't want too tight binding because it's not useful to have a very tight binding, since the oxygen needs to be released then to myoglobin and to the muscles to perform function, right? So that explains why we only want the strongly binding hemoglobin in the organisms or in the fetus that really need a high binding affinity. We don't want high binding affinity everywhere. Yeah, just stressing the balance of always function, so it's never quite as simple as it looks in the first place. So what do you think then in terms of classifying organisms from the more simple to the more advanced? Do you think proteins are going to be more complex in higher organisms? So if you compare bacteria with a human, do we have more complex proteins, and in which way? Bigger, smaller, more complicated? No idea? Well, guess who says bacteria have bigger proteins? Yeah? Who says bacteria have smaller proteins? Okay. Want to have a debate?
I don't know, I just took a stab in the dark, but I might be completely off now, but I thought maybe they have large proteins that can perform more functions because they're small organisms. I don't know.
Yeah? Good reasoning, but unfortunately it's the other way around. But it could be, it's a bit the story actually of biology. You can defend a little bit many points actually. What did you guys think about why bacteria would have smaller proteins? Or more simple? Just random?
It just felt reasonable that because they are so size smaller.
They have what?
The size is smaller than
Yeah, so the idea is making protein costs energy, right? So you pay the price. So if you need a protein that needs to perform a complicated function, it needs to be big. then you have to pay an energetic price. And bacteria are usually really quite simple, actually. I'll show you an example soon of this. But what we see, in fact, that's quite interesting is that, yes, the proteins of eukaryotes, mammalians specifically, they're usually bigger, more complex, but they're more complex just because they add domains. So it's not that the folds are more complicated. It's not that you have, weird many helices intertwined with beta sheets. The domains will be very similar in bacteria and in eukaryotes. But the way they are arranged makes more complexity in eukaryotes than in bacteria. And then the other thing you might have heard, or maybe not, is that eukaryotes can also have more complicated proteins because they can undergo post-translational modification. Have you guys heard this term before? No? So, no? Yeah? Maybe? So now we go from DNA to RNA to protein, and then you guys have stopped here at this point, right? But in fact, in eukaryotes, there is something that is called post-translational. So translation is from RNA to protein, and it's decorating the protein. So for example, glycosylation is a famous one. This means there are enzymes that add sugars to already built proteins, and this creates complexity. So now you know eukaryote proteins are more complicated, but only because they have more domains and also because they're more decorated, which means that the folds in general are going to be very similar in all the organisms. And this kind of goes towards this idea of putting pressure on from physics, right, to design what is possible or not. So this is the example I wanted to show you, and we already actually talked about this in the lecture on membrane proteins. So this is an ion channel. Functional principle is that it needs to conduct ions to the central pore, and it has a hydrophobic core that's exposed to the membrane. Yes? And compare these two, bacterial to eukaryotic. The central part is very similar. You can tell, right? You can see these pore lining helices, same orientation. And the second helix here, same orientation. So basically, this protein, if you cut it here and remove this part, it's the same as this one. And it's a tetramer, so I'm just showing you on one monomer, but the same thing is repeated 4 times. Yeah? So this is really this decorated by these domains. Do you remember what this was from the membrane protein lecture? Okay, this means voltage-gated potassium channel. This is a potassium channel, not voltage-gated. In bacteria, it's actually gated by pH. But this is gated by voltage. And so the role of these domains is to sense voltage. And bacteria don't need to sense voltage, but they need to sense pH. But higher organisms, they need to transport electrical signals in their nerves, muscles, heart, and so on. And so they need then to have channels that can do that. But the organism pays the price because it's more expensive to synthesize this. But it needs it to live, so then it pays the price. So this is just a simplified story because it turns out that bacteria actually have some voltage-gated ion channels and nobody knows why. But the simple story is this, and it gets repeated many times. So now we just jump quickly to the question, how fast does evolution take, natural selection? How long does natural selection take? Typically, people say millions of years, right? That's what you have in mind, like, monkeys turning into humans over very long periods. But this is just a study that shows you that evolution of structures can happen really quite fast. So it might not be a dramatic change like adding domains. But this shows that this is actually taken from a study on fish, cod, living in different areas. And the Hudson River, that I think goes through New York City, is extremely polluted. It has a lot of these so-called PCBs, which is a pollutant. And if you look at the population of proteins inside of cod, you can see that they're very different depending on whether they live in the Hudson or not, and there's kind of a gradual change over regions that are more or less polluted. So the cod has managed over a few years, right? Because we started using PCB in the 50s or something, not a million years ago. So if there's really a high pressure, that means that this pollutant is going to kill the fish. The one fish that manages to escape this pressure really can transmit its genes very fast, right? And so then you have this change of whole populations, not individual fish, over quite small periods of time. So to give you an idea of how fast this can go. If the evolutionary pressure is much smaller, then you might have things that happen over much shorter periods of time. Yeah? Okay, so now we come to our big question of the day. How does nature seem to recycle always the same patterns, although there are a wide variety of sequences? And this comes back to structural stability. And you know, structural stability comes from what? Hydrogen bonds forming, right? Hydrogen bonds from the backbone of the amino acid chain that self-organizes to create hydrogen bonds between, in alpha helices, the amino acid at position I and the amino acid at position I+4, right? Or in beta sheet, a straight strand with the polar groups from the backbone pointing one way, and then another sheet coming down and hydrogen bonds forming from adjacent strands. Yeah, that sounds familiar? So this is the basic reason for structural stability. And this means that loops, coils, cannot exist inside of the protein because they have hydrogen bonds that are exposed, right? So they need to be in solution because solution can, water, meaning can come and create hydrogen bonds with the exposed backbone chains. And this also explains why membranes have a lot of helices. This we talked about already in the membrane protein lecture a while. So the additional thing that occurs from this is that if you take a single beta strand and you pair it on one side, the other side is not paired, this one has to be exposed to water, right? It cannot be exposed to a helix, for example. That would cause too much penalty in terms of energy. And this is also the reason why helices and sheet tend to be separate. So imagine if you have one sheet, a helix, one sheet. Is that more stable or less stable than a sheet, a helix? You get it, right? I'm just kind of repeating, but we're good. And so if you look at the ensemble of structures, then you'll start noticing that there are, in fact, very few defects in protein structures. And this is because because defects are costly. So remember burying a charge, how much that costs, this sort of thing, or removing one hydrogen bond. Yeah.
When you say defects, you mean defects in the protein that might affect their function such that the organism...
No, I mean defects in terms of thermodynamic stability.
Right.
So I mean having a break in this helix.
Sure.
Or I mean having a beta strand of which the edge is close to an alpha helix.
Right.
This is what I call the
so
defect.
So basically is something that would deviate from what is physically or thermodynamically advantageous.
Yes, absolutely. So there will be exceptions, of course, but if there are exceptions, you have to pay an energetic price. That's the basic. So we can think about this in terms of layers at this point. And as we talked about before, actually, by just looking at the different folds, you can see that usually these proteins, whether they're alpha, beta, or alpha and beta, they're organized in layers. So one layer would be one sheet, with all the side chains pointing up and down, right? And a layer of helices would just be helices lined next to one another. This is not very useful for function because it's like a piece of paper, right? Like you can write on it, but you can't really do anything mechanical with it except if you start folding it, right? And so that is why 2 layers is actually the most prevalent thing, because with 2 layers, now you can start binding something in between, right? For example, or you could bury some kind of interface that allows to do something interesting. 3 layers exists also. The Rossmann fold is very famous. That's what's represented up here. Then you have a beta sheet in the middle and helices on the side. This can create 2 different functions in the same protein. So you would have, do you see this? The sheet with the helices on both sides. So you can create something going on in between these 2 layers, and then something going on also between these 2 layers. So it just makes it kind of more interesting. 4 layers is extremely rare. Why is that? What do you think? Do you remember the proportion of hydrophilic, hydrophobic amino acid in nature in general? No? 50-50, roughly. So we have about equal numbers of hydrophilic and hydrophobic. So what would happen in 4 layers then? Four layers would mean a hydrophilic side, a hydrophobic side, right? Then hydrophobic, hydrophobic, then hydrophobic, hydrophobic, and then hydrophobic, hydrophilic. Yes? Did you follow? Yeah? So then we would want to bury a lot of hydrophobic residues. And since we have about 50-50 hydrophilic, hydrophobic, a lot of protein sequences won't fold into 4. It doesn't mean you have to imagine sequences being random, right? We just pick an amino acid and throw it in from the overall population. So this is why we don't have 4 layers. And this is also why we don't have more, 5 or so. And so this means that large proteins, like we just said, once you reach a large number of amino acid, then they need to be subdivided into subdomains, because we never have 5, 6 layers. So if you have a very long protein chain, then it's going to arrange into a domain that will have 2, 3 layers, another domain that will have 2, 3 layers, and another domain that will have 2, 3 layers. And this will create a bigger protein assembly. So like I said, now what we're going to ask to try and understand these folds is to ask which sequences can fit into a given fold. We've just put boundaries on the folds, right? We know that they have to fold in kind of specific assemblies with a specific number of layers and in helices and sheets. So this means that not all the sequences will be allowed. So we're reversing the question. We're asking which sequence can fit into a certain fold. And if you think about it in terms of probabilities, we said defects are allowed, but defects will require something special to stabilize it, right? And if they require something special, it means that not a lot of number of sequences will be able to fit this fold. So if we think about it, the more defects, then the fewer sequences can be accommodated. And this is then, it decreases the likelihood that this will happen if you consider a number of random sequences. This is why we see them less. Is that clear? This is kind of really the central point of this first part of the lecture. But we're going to talk in a little bit more details about like the physics. We'll make models to try and explain this in a minute. Before that, I just want to show you one actually motif, but there are many like this that just keep coming up again and again. And look at this. So this is called a Greek key connection. You can see it's 4 helices that are colored here. that are oriented anti-parallel. So this is how it's stable, right? And then you can see that to make this happen, you need to have a very short loop here and a kind of long loop in red up there. This is called Greek key motif. And in fact, somebody, I forgot who, noticed that this was very similar to the patterns you see on Greek vases. Do you think this is a coincidence that nature has the same kind of pattern than Greek people a long time ago?
Yes.
It's a coincidence?
Yes.
Yeah? Okay. So it could be. We don't know. But think about how you can envelop a volume in a line that doesn't crash back into itself. There's actually not many ways to do this. So this is pattern filling, right? It's like something that artists like to do. And if you think about you're going to put one line and you want to fill some volume and the line can't cross itself, then this is actually a kind of interesting way to do it. And this allows in nature to bury hydrophobic things, and in art it allows to have an interesting pattern that folds onto itself. So I think folding is, kind of interesting. By the way, we also fold, napkins and sheets to try and make it take less space. This is how you make a line take less space without it crossing. But what I want to highlight here is that this is putting restrictions on the sequence, right? Because you need to have then residues that like to be in beta sheets. We saw in the bioinformatics class that it's not all residues that like to be in beta sheets, but there's a decent number who do. And you need to have residues that like to be in a tight loop, and residues that like to be in a long loop, and residues that also don't want to be alpha helical, right? So all of this together makes it possible for this motif to happen. But we should consider how many possible sequences can fit into this kind of motif. And this is what we'll do. So like I said, we're considering these random type of assemblies. The world of amino acids is divided into two types of amino acids, very simple. Black ones hydrophobic and white ones hydrophilic, just as our model. And like we said, there's about half-half in the universe. And then how can we organize them? Well, if it's a membrane protein, then the assembly is the chain is gonna look like many hydrophobic in a row, and then mostly hydrophilic, and then mostly hydrophobic in a row, and then mostly hydrophilic. This is to create the transmembrane helices and the loops in between them. Yes? Fibrous proteins, they have a mix of hydrophilic and hydrophobic, but they are really organized in repeats, because you remember these things with the Alzheimer's? Remember fibril proteins that assemble together to form fibrils? I think that was in one of the early lectures. You also talked about silk, maybe, or yeah? So this was, if you take one repeat, it doesn't form anything, but then if you start having a repeat next to it, and next to it, and next to it, then you have something that starts appearing, some structure. And so this is for fibrous. Globular proteins, they look random, is what you see up there. So they're not random. We know they're not random because they have to perform a function. But if you just look kind of coarsely at the sequence, you wouldn't be able to notice any pattern. It would just look random. And from these random sequences, we're going to talk about what type of structures can arise. So before we go into this kind of model, I want to ask quickly why we think defects are rare. So related to this defect problem, if you look into the type of folds when you arrange helices together, you see that anti-parallel helices are very common, and parallel helices with a long loop in between are really uncommon. So this would be called a defect, and this would be called a regular assembly. And if you have more than 2 like this, you have 4, then the crossing of these type of loops is really very rare. And it's kind of mysterious why, in a way. Because, okay, if this happened, right, maybe this forces this loop that is maybe 2 amino acids kind of closer to the membrane surface, or to the protein surface. And this looks like it would cost a penalty of maybe 5 to 10 kcal per mole because it's 2 hydrogen bonds that you're mismatching. So it's a little bit mysterious why. This doesn't seem big in the, like a big number in the big scheme of things, right? Imagine you're folding an entire protein. How many hydrogen bonds is that? So what's the contribution to energetics from all these hydrogen bonds? Why cannot we afford to have a small defect? This is a question we'll answer in quite a lot of details in a minute. Oh yeah, that's another side of things that put constraints on the ensemble of structures. And this is the notion of, amongst all these folds that have 2 or 3 layers, there are going to be some that accommodate more diversity than others. So I don't know, I don't actually have an example for this, but imagine if you have a beta sheet and 2 helices on top, then depending on the size of the beta sheet, maybe the 2 helices can be in many places, right? Whereas if you have 3 helices or 4 helices on these beta sheet, then you can't really, there's no room to change this. So some fold that allows more possibilities than by the principle of entropy just allows for more entropic gain upon folding, and so this is why some folds are also more prevalent than others. Side point, it doesn't matter. So this relates to this question of why we don't see defects too much. We know how we can measure probability distributions. You've learned this, I think, previously in statistical mechanics and in this class. Probability distributions are governed by the Boltzmann distribution. Yes? This I know you know because you handed in very nice reports. So can we describe these structural elements in a protein using this formula? The book argues we can't. So even though we observe many folds of low energy that are very prevalent and very few folds of high energy, this looks like it's described by something like the Boltzmann distribution. Yeah? But in fact, the Boltzmann distribution has built into its construction that you need to be able to exchange between conformations back and forth through a principle called detailed balance, right? So for example, particles go from being ordered to disordered to reordered to disordered, and the number of times it's going in one direction has to be equal to the number of times it's coming back. When we think about the structural ensemble of structures, we're not exchanging between, having, let's say, this beta sheet 1 helix here and 1 helix there, and this beta sheet with 2 helices there. This doesn't happen in nature. It happens over evolution, but physically it doesn't happen. Yeah? So we can't, in fact, use the Boltzmann distribution to think about this. But there is a little bit of mystery because, like I said, the ensemble seems to be governed by something that is exponential and related to energy. But it won't be the Boltzmann distribution, and we'll derive this afterwards. So this is the core principle, the multitude principle that governs which folds exist. And it says that the more sequences that can fit a given architecture or fold without modifying its stability, then the higher will be the occurrence of this architecture in native proteins. So we're based on this physical principle of having half-half hydrophilic and hydrophobic amino acids, and we're building these random sequences. And the more different types of sequences that will be able to fold into a specific fold, and that is stable, this will then say that this fold happens a lot in nature and this other one doesn't.